Thank you. Thank you, Harry, for introduction. And yes, today I'd like to introduce a new sensor from the lab, which is a graph sensor for imaging endocannabinoid in vitro and in vivo, especially in brain. I will start from introducing this plant, cannabis. Actually, it has been used for more than 5,000 years for both medicinal and recreational purpose. And this plant has this effect because it has these molecules we call cannabinoid, for example, the tetrahydrocannabinol, THC. These molecules can actually hijack our body's endocannabinoid system to exert their function. So what is the endocannabinoid system? This system composed of the endocannabinoids, the endogenous cannabinoid in our body, mainly two molecules, 2-EG and AEA, and also the receptors, CB1 and CB2. CB1 is mainly expressed in the nervous system, while CB2 is mainly in the immune system. So in addition to the ligands and the receptors, there are also many other proteins that regulating the production and degradation of the eCBs. And in the nervous system, people believe that they function as neuromodulators. And interestingly, distinct from other classic neurotransmitters that are released from presynaptic terminal, the endocannabinoids are actually produced from postsynaptic set and retrogradely modulate the presynaptic transmitter release. So it's kind of a unique lipid modulator. And basically, this eCB system function in almost all brain regions and play important role in many processes, including learning, memory, sleep, motivational behavior. And also the system is tightly linked with different diseases, for example, epilepsy, addiction, pain, and neurodegenerative diseases. So although we know many about the function biochemistry of the endocannabinoid, however, a key question actually still remain to be answered. That is the spatial and the temporal dynamics of the eCBs, especially in the brain. So people guess that it functions in the time scale of seconds and diffuse to tens of micrometers. However, this we do not have direct evidence for this. So we developed a GPCR activation-based sensor for endocannabinoid, short for graph sensors. We taking advantage of the endogenous sensor for eCBs, the CB1 receptor, and we find previous studies find that activation of the receptor leads to the conformational change. And we thus insert a circular permutated EGFP into the receptor. And thus we use the fluorescence intensity to report the ligand binding event. So thus this is the principle of the sensor. The sensor is genetically encoded, so it can be expressed in specific cell types to report the cell types, to report the signal with cell type specificity. And it has relatively high, in principle, has high sensitivity, specificity, spatial temporal resolution. And also it is easy to use, just like GCAM in calcium imaging. So after several rounds of screening, we got the second generation of the sensor. We named it eCB 2.0. And when expressing HEC cells, it shows good membrane trafficking and the response to both AEA and 2-EG in a concentration-dependent manner. We also generate a mutant sensor by introducing one mutation in the ligand binding pocket of eCB 2.0. This mutant sensor no longer responds to endocannabinoid, so it is a negative control. And the response of the sensor actually is quite specific because it do not respond to other neurotransmitters or neuromodulators, and the response can be blocked by AM251, the specific inverse agonist for CB1 receptor. In neurons, the sensor traffics well to both axons and dendrites and even spines, so the sensor is able to report eCB signals throughout the neural membrane. And similar like in HEC cells, the sensor can respond to AE and 2-EG in a concentration-dependent manner in cultured neurons, and the eCB mutant sensor actually show no response in all concentrations we tested. Next, we asked whether we could report the endogenous eCB from neurons. So, and also to test whether it's able to combined with other sensors, for example, the red sensors, we express ECB 2.0 with a red glutamate sensor together in culture neurons. And the electrical stimulation could evoke both ECB signals and glutamate transients faithfully. And now similarly, we could also detect the ECB signals and the intracellular calcium signals using a calcium dye. And also, consistent with previous study that the ECB production is calcium dependent, and we see a good correlation of the peak ECB signal with calcium signal. And also interestingly, in culture neurons, even without electrical stimulation, we could see spontaneous ECB dynamics that are relatively local and small spot. The diameter of this local transient is about 11 micron, so indicating it's kind of a local signal. And it's probably for the first time we are able to have this relative high spatial resolution. And temporally, the spontaneous signal are also actually different from the electrical stimulation evoked signal. So indicating this spontaneous signal actually shows different releasing pattern compared with the stimulation evoked ones. And then we also validate the sensor in a more physiological relevant context in the acute brain slices. So this part is in collaboration with Dave Lovinger's group at NIH. We express the sensor in dorsolateral striatum, and electrical stimulation could evoke a robust ECB transient recorded by photometry recording. And the signal actually is stimulation pulses number dependent and frequency dependent. And this is a quantification data of the peak responses and the rise and decay kinetics. And then importantly, the signal we observed is specific because pre-treat the slice with AM251 or using the mutant sensor, we could not see the stimulation evoked signals. We also found similar result in hippocampal slices, and stronger pulse stimulation evoke larger signals. And this, again, the signal is specific because in the presence of AM251, the same stimulation no longer evoke the responses. So next, we want to ask whether we are able to use the sensor in vivo to report the ECB signals in living animals. So we express the ECB 2.0 sensor together with Emcee in mouse basolateral amygdala. And again, we find a good expression of both fluorescent proteins, and the BLA is a key brain region that mediating the aversive responses. So we give the mouse a foot shock and record the signal using fibrophotometry. We could see a time-locked ECB signal increases upon stimulation, but no response in the Emcee channel, so indicating the specificity of the signal. And in the same mouse, we have repeated trials, and the response are stable, and also the repeat are stable in different animals. So this part is in collaboration with Boley's group at Cold Spring Harbor lab. And also previously we showed that the sensor is able to report ECBs in hippocampus. So next we want to know whether we can see the in vivo signals again in hippocampus C1. So express the ECB 2.0 sensor together with a red calcium sensor GAG1A together in hippocampus C1, and using a 2-photon microscopy, we can see the expression of the both sensors. And when we put a mice on the treadmill, we can see both on calcium increases and the ECB increases when the rat starts running, and the stop of and the decrease of both signals when the mouse stops running. And lastly, we want to know whether the sensor can be used to report the ECBs in some disease conditions. So in this case, we using electrode, we use electrode to stimulate the contralateral hippocampus and imaging the ECBs in the other side. And we can see that when induced when using the kindling protocol to induce the seizure, we see both increases of calcium signal and ECB signals. So, and as a control, the mutant sensor actually did not show any signal. And interestingly, after the seizure, we usually could see a spreading wave across the CA1 region. So this is a video. It doesn't work. Anyway, we could see actually a spreading wave across the CA1 that of both calcium signal and ECB signal. So these relatively large signals could demonstrate the sensitivity of the sensor as well as the spatial resolution of the sensor. Okay, so in summary, we developed a graph sensor that based on human CB1 and cpEGFP, and we characterize the sensor in both culture cells, neurons, and the brain slices. And finally, the sensor is able to report the ECB dynamics in vivo, both in BLA and in hippocampus, under physiological and pathological conditions. So in the end, I want to thank my advisor, Dr. Yulong Li, for guidance, and also many collaborators in the lab and in other institute. And you can follow us on Twitter or lab website for information and reagents. Thank you.
Thank you, Al. This is really beautiful work. And if you have any questions for Al, please use question and answer panel down to ask your questions, or chat panel to ask your questions. For this sensor, as far as I see, this is just the second version of it, and it's already started. Do you think there is any range for making the signal to noise ratio better? Because
the current version of the sensor, actually in culture neurons, we could reach to like 6-fold to 8-fold responses. And for in vivo conditions, it depends on the depends on the protocol, I think, and also the recording conditions. So I think for the current version, ECB 2.0 is already enables to show the in vivo responses. And also we are working on to improve the response and the dynamic range of the sensor. And also we are working on to expand the color spectrum of the sensor. So maybe also easy for use combined with other sensors. Yes.
Okay, thank you very much. Again, if you have any questions for Al, please use the question and answer panel to ask him. We will move to our next speaker, but he will be around to answer the questions. And now we will move to our next speaker.